Okay, class, we are continuing on with looking at inside the heart. These are the papillary muscles pulling on the cardiac tendinate, which pull on the valves to keep the valves from bending back the wrong way, to hold so that the blood does not go back the wrong way, regurg. Here again is looking at these papillary muscles, cardiac tendinate, and the valves. Again, the various valves. Here's a sequence of events that you can read on. So basically you can read it. Blood goes in. So let's say this is coming from an atria. The blood will then go in because the atria would have caused the blood, enough pressure would have built up in the atria to cause the blood to go into the ventricle. When the blood goes into the ventricle, then subsequently, eventually the ventricle will contract. When the ventricle contracts, it would push blood up because this chamber would be somewhat filled, not completely. Blood would then push up and the pressure would attempt to push the valve the wrong way. But these papillary muscles and cardiac tendini hold the valve so it doesn't go the wrong way. So then what would happen is, once enough pressure builds up, since the inlet valve on the right would be the tricuspid, on the left, the bicuspid. Since the inlet valve is now closed and the ventricles are still contracting, pressure is building up quite a bit. Finally, there would be enough pressure to pop open the valves. If it's coming from the right ventricle, it would pop open the pulmonic valve. If it's coming from the left ventricle, it would pop open the aortic valve. And then the blood would go into the aorta from the left side and the pulmonic from that side there. So that's essentially what's happening there. Here's showing some abnormal valves. Here's a prolapse where the valve goes back the wrong way, mitral valve prolapse. It could be enough to have a regurgitation of blood. A stenotic valve is a tight one. That means it's really kind of the opposite of a prolapse. It's that the valve is too narrowed and tightened. So those are some abnormal valves. Here's looking at valve replacement surgery. Sometimes I just threw that in so you could glance. There again, replacing heart valves. Valve replacement, doing it without having to go in all the way. Without an open valve. This is a percutaneous. So that's where we are there. Okay. Now we're looking at the histology and physiology of cardiac muscle. Okay. What are the requirements of cardiac muscle? One is considerable endurance. What do I mean? The heart is beating over 100,000 times a day and for the rest of your life. The next thing is both atria must contract at the same time in order to squeeze the blood effectively into the ventricles. And then both ventricles must contract at the same time. Because if one side contracts before the other, you'll just flop the blood around by bending the interventricular and interatrial septum. The endurance then basically involves that, comparing this to skeletal muscle, that the cardiac muscle cells are smaller than skeletal, giving them what we call a better surface to volume relationship. That means that the surface area is nice and big where oxygen and nutrients can enter. More myoglobin. I don't know if you remember, myoglobin is the red stuff in muscle. Myoglobin is the brother of hemoglobin. It holds on to oxygen. So in muscle with a lot of myoglobin, it holds on to more oxygen, giving it more endurance. A slow ATPase. When energy is released, some energy will go away as heat. The slower you release energy, the more energy will go towards useful work versus heat. The slower you release it. So, because energy cannot be created or destroyed, it's going to leave. So if you took a match, lit up your gas tank, you would release all the energy from there. but your car would fly, but it wouldn't go as far as if it went the proper way, because the proper way would allow it to be released more slowly. So cardiac muscle has an enzyme, a slow ATPase, which causes ATP to be broken down slower, which gives more endurance. Heart muscle also uses almost exclusive aerobic metabolism, but it can burn lactic acid. It can burn lactic acid, and it has lots of mitochondria. Okay, so here's a comparison of cardiac muscle. Now right here, these are the three types of skeletal muscle you should have studied in ANP1. Slow oxidative, fast glycolytic. The slow oxidative would be your endurance muscles, like your postural muscles, leg muscles. Fast glycolytic would be like your, let's say your biceps. They move fast, but they tire quickly. So what I'm saying is that cardiac muscle even has to have more endurance than slow oxidative muscle. So therefore, it would have more myoglobin and various other factors it would have. Okay. So under basal aerobic conditions, 60% of the energy comes from fatty acids and triglycerides. Remember, in most cells, carb is the main. But in this cardiac, it loves lipids quite a bit. Okay. Atrium and ventricles must contract as a single unit. Okay, here's the situation. Cells can only get so big. Cells can only get so big. Because cells can only get so big, you can't make one big muscle cell to surround the atria and one big muscle cell to surround the ventricles. If that was the case, then you definitely would have, if you could, both atria contract at the same time, both ventricles. But you have to break the cells up into the muscle cells into small units. So what happens is, though, you still want to try to make it act as one, though it is not one. So in other words, you'd like to have it act as one muscle cell, even though it's a bunch of muscle cells. So what is attempted to be made is a syncytium. A syncytium is, though you're many, you're acting as one. Though you're many, you're acting as one. So the cardiac muscle cells have to be hooked up in such a way to act in a syncytium fashion. Okay? In order to do that, first of all, the cardiac muscle cells are branched. They're branched. That means it's almost like a chain letter. That means the charge will go down several channels like a chain letter. Each branch hooks to another cardiac muscle cell. So it's almost a geometric progression. Once the signal travels down several branches, it goes much faster. The cells are hooked together by gap junctions, AP1. Gap junctions, the intercellular junctions, allow the impulse to travel through the intercellular spaces much faster. Okay, so the fact of the branching and the gap junctions give the muscle a syncytium. So though there are a bunch of cardiac muscle cells in the atria and a bunch in the ventricles, there is a syncytium for the atria and a syncytium for the ventricles. When you look at the microscopic anatomy of cardiac muscles are striated, just like skeletal muscles. They are short. They have the T-tubules, but for contraction, most of the calcium comes from outside the cell, and they do have mitochondria. Here's a comparison of skeletal, cardiac, and smooth. Now, I encourage you to look this up. Cardiac muscle cells are hooked by intercellular junctions. They have what we call intercalated disc. Okay, the intercalated disc are a light microscope structure. It's an interspersion between desmosomes and gap junctions. The gap junctions allow ions to pass quickly, the sodium potassium. The desmosomes prevent, when the contraction occurs, for the muscle cells to split apart. So these two features here, desmosomes and gap junctions, can only be seen under the electron microscope. But what they actually are under the light microscope, you would see intercalated discs, because you could not resolve and see desmosomes and gap junctions. The heart muscle behaves as a functional unit called a functional syncytium. Okay? So this is a look at this. Here would be cardiac muscle. Here would be the intercalated disc, but the intercalated disc under electron microscope are an interspersion of gap junctions and desmosomes. You can see that the muscle is branched. So read back on these features I've given you. Here it is again, branched. Here's again. So read back on those features and in the text, and I think that you will be able to understand this. Thank you.